así o le va a prestar su teléfono?
Ah, me va a prestar su teléfono, sí. Sí, o él me las va a decir. Ya vemos. Well, I think, creo que ya es tiempo de empezar. Qué lástima que hay tan poca gente. No duden que se van a encontrar muy buenos datos y información. Seguramente van a aprender todos, o vamos a aprender todos. Bueno, pues con la audiencia que está ahora, tengo, I'm going to introduce you in Spanish, that's fine for you. Tengo el honor y el gusto de presentarles al doctor Dan Jarozs. Él es ahora profesor asociado de dos departamentos, del Departamento de Química y del Departamento de Systems Biology. Del Departamento de Química y Systems Biology y del Departamento de Desarrollo de Developmental Biology en la Universidad de Stanford. Hizo su licenciatura en la Universidad de Washington en el área de Química y Bioquímica y después su doctorado en el Whitehead Institute en MIT en Química Biológica o Fisicoquímica. Y posteriormente hizo su postdoctorado en el Whitehead Institute en Genética y Biología Celular. Eso fue en el 2012 y como les dije, ahora ya está en Stanford como un investigador independiente. También pertenece a un centro o red que se llama Sarafan Chem H, que es muy interesante porque es una red de investigadores que están dedicados a tratar de resolver problemas relacionados con enfermedades y con envejecimiento. Y pues es una forma de colaborar con diferentes experiencias de los diferentes miembros de esta red. Durante su postdoctorado, el doctor Jarozs estudió la chaperona molecular HCP90. ¿Y ahora por qué me están oyendo? HCP90 y su relación con causar variación genética para producir nuevos fenotipos. De ahí surgió su interés en estudiar proteínas que conocemos como priones y que sabemos ahora, justo tiene que ver con el título de su plática hoy, que de alguna forma se regulan o se comportan como componentes de una regulación epigenética, ¿no? Pero a nivel de proteínas. Los descubrimientos que ha hecho alrededor de los priones han sido realmente relevantes y ahora pues maneja diferentes temas relacionados todos de alguna u otra forma con priones, pero también con enfermedades relacionadas al envejecimiento y otras como cáncer. Y sus contribuciones a nivel de biología básica también han sido realmente interesantes e importantes porque le da un sentido a estas proteínas que originalmente se habían relacionado principalmente con enfermedades, pero ahora tienen un sentido más biológico desde el punto de vista evolutivo y desde el punto de vista funcional, que supongo que nos va a platicar hoy. Entonces, le agradecemos mucho al doctor Jarozs que haya aceptado la invitación de la licenciatura en Ciencias Genómicas para estos seminarios de fronteras en genómica y pues le doy la palabra. Thank you very much for accepting the invitation and the auditorium is yours.
Thank you so much for this. Okay, good, better. Great. Thank you for the kind introduction and for the very wonderful hospitality during the visit. And yeah, I'm excited to tell you today about some of our work in the areas that you just heard about and others as well. Okay, so for much of my training, I have been captivated by this kind of paradox that occurs in life and evolution. If you think about how you look now, for better or worse, it's pretty similar to how your parents look, right? And that's because life devotes a great deal of attention to making sure it gets information transfer right from one generation to the next. And we study that in the sequences of genomes. We study it as cell biologists in how chromosomes are transmitted at cell division, et cetera. And again, as a result, there can be extraordinary transmission of traits within families because that information is transduced so effectively from one generation to the next. But we also know that life requires extreme adaptability, right? It's very tough, right? And so that requires change in response to a challenging environment. And this creates a difficulty, right? Because how do you reconcile the two competing demands, that you need to have everything be the same over and over again, and yet you also need to change in order to ensure long-term evolutionary survival? Indeed, how does life balance these needs, right? One way is stress responses, right? And we've all heard about ways in which organisms change their gene expression profile, either transcriptionally, post-transcriptionally, or in other ways, in response to stress, right? And that can have a huge effect on biological outcome. So here I show you just some examples of data, both in yeast and in plants, of how much that response gives you in terms of survival. So in each case, on the left, are organisms that were heat shocked directly. And as you can tell, they died, okay? The ones on the left, all that is different about them is that they were given a mild treatment at a slightly elevated temperature for a little while, and then they got the heat shock, right? And in that time, they turned on the expression of all kinds of genes that are going to protect them when they get the more severe stress later. So this is an incredibly adaptive response that is conserved from bacteria to us. And I want to emphasize here that there is no genetic change. If you take these guys and take their progeny and do the same thing, and you didn't pretreat them, they're going to look exactly like the ones on the left. Okay. We also know that organisms can take a different strategy, and in response to stress, they can induce mutagenesis. We see this a lot in the context of DNA damage. And in fact, my thesis was devoted to this in my PhD. And it turns out that this process can actually be very strongly regulated. Some organisms can, in certain contexts, upregulate their mutation rate 1,000-fold. And in fact, there are specific cases in which they can target it to places that are useful for them. And this also is an important mechanism of adaptation. Yeah. So we usually think of adaptability as a good thing, but of course, in the context of our health, it's often a huge problem. So in microbial infections, this is one of the chief causes of treatment failure, the acquisition of resistance that comes from evolution, right? And at least in the US, probably here as well, we read all kinds of headlines like this one about concern that in a shortish amount of time, we may reach a circumstance in which there won't be drugs that are available to treat these sort of infections. And in fact, the impact of these are extraordinary. This is data from the World Health Organization in 2019. As you can see, this resistant infection is a greater cause of death than many things that would scare us far more if we were to receive the diagnosis. Okay, so. And that is actually an underestimate because you only think about microbes. In the context of cancers, right, that are filling up many of those other bars, in fact, there too, right, one of the greatest causes of treatment failure is also the acquisition of resistance. And kind of a new frontier in therapy is trying to predict what direction the tumor will evolve in and then treat sequentially in ways that kind of block each of those pathways. Okay. And I realize this all sounds like doom and gloom, but it will be interesting. This will be more hopeful later. The problem is made worse by us, right? So, the miracle of our modern life comes from all kinds of chemicals that we make, and many of them are secreted into the environment, either intentionally or unintentionally, by us. And what this means is that there are low levels of many of these drugs that we want to use and not have resistance against in the environment. And usually we don't think of it as being a big deal because it's not so much, right? It's very far from the therapeutic dose that we might use. But actually, sometimes it can matter. Okay, okay. So of course, evolution also does great things, right? It sort of creates all kinds of beautiful forms and functions that we observe in nature and with those who work with us. And I live in kind of a department that is obsessed with trying to understand at a very quantitative level how regulatory and functional circuitry work inside of organisms. And people like to see kind of this type of a diagram and build a lot of partial differential equations to understand it. But of course, like the wiring of this is affected by many things, including variation in each of our genomes, in our sort of life history encoded in epigenetics, as well as stress and other things that we face on a daily basis. Okay. And this kind of thing makes us wonder, like, are mutations the only mechanism that matter for adaptation? And it turns out that theoretical biologists have thought a lot about this question, and they aren't always. So really what strategy you want to adopt depends on the nature of the stress. If it's going to be pretty reproducible relative to the lifespan of the organism and transient, you want a stress response. You don't want to make a mutation to turn on a response all the time. You want to have on and off. Okay? If you have a trait or a change in the environment that is instead less frequent than the generation time of the cell and maybe less predictable in terms of what its duration is going to be, then you actually favor an epigenetic response that can be both heritable but also reversible. And indeed, if it's a permanent change, you favor a mutation-based mechanism. So what I will tell you about today is kind of one of the middle mechanisms that is a very unusual one that was found as a result of a disease that baffled molecular biologists, but also shepherds, veterinarians, and even people in the House of Commons in the UK for centuries, because it was based not on a pathogen that came from the outside, but rather from within. And this is, of course, the classic prion problem. And it is a very different situation of folding than what we are used to, where we think of the primary sequence of a protein encoding a structure and a given function, and a different sequence encoding a different structure and different function. In this case, it's something very different, where large numbers of proteins don't look the way that we were taught in school. They have ordered domains, usually, although not always. And then they have large regions that don't form a single fixed structure in solution. And these can do really crazy things. And many of you are interested in this question. Large fractions of our proteome are disordered. Right? And it is, has long been something that people ignore. As a biochemist, I was trained to immediately try to identify what those regions were and remove them because they didn't matter for function and they caused all kinds of problems with the behavior of your protein. Okay. And those sequences can endow a protein with many properties, and sometimes it allows the protein to adopt different conformations, but not just to adopt that, but to make this conformation that can template, that is heritable. And classically, we're used to this in the context of a fiber, where the ends can basically template new monomers to add. They undergo a structural conversion, and then it keeps growing and growing and growing. And that creates actually a mechanism of inheritance. But we have shown that all other types of structures can form that have the same properties, that basically are able to be a protein replicon, an element of inheritance. So again, this story begins with disease in sheep that really concerned these shepherds. And if you read the accounts from the House of Commons, you will see that they described a very unusual pattern of inheritance where one sheep might have been sick 18 months before, nothing happened, and then the whole flock would suddenly succumb to it, right? And they couldn't figure out, they do all these things that they knew should block the transmission of other types of pathology, and they did not see anything. And it was a problem of interest to scientists within the 20th century. They could show that you could transmit in various ways that didn't make sense. And it was actually Tikva Alper who had the first hypothesis that it could be a protein, but she was not able to show this. And then Prusiner, of course, famously showed that this was from the conformational conversion of protein PRP into a templating form. Okay? But it turns out this property is not unusual at all. It's present throughout life. And one of the most helpful examples in terms of our understanding of its basic biology comes from the example on these two plates of yeast that were also discovered in the 70s. It was this phenotype where the red cells that are red because you've introduced a stop codon in a specific gene, they become white, right, randomly. You can see that happening here, for example. If you pick that white cell and then you streak it again, it's white. Okay? So it's a heritable change. And you can see all of the progeny remain white. But if you sequence them, they have exactly the same genome. Right? There's nothing that is different about them in that way. If you pick these cells, the white cells, and you propagate them on a plate that would contain a little bit of guanidine hydrochloride, they will go back to being red. Right? All kinds of weird things happen with them. Okay? And what's different about them is that they have a different proteome, right? But not one that you would know if you did proteomics on these strains. What they've done is taken a single protein and reorganized it into a structure that can, again, template. And you can see that if you tag that protein, which is part of the ribosomal release machinery with GFP. Ordinarily, it's present throughout the cell, but in the white cells, you see the emergence of these puncta. They come from bundles of amyloid fibers that are organized together. They look almost like a honeycomb. It's very beautiful. And in fact, that protein alone, if you just purify it, has the capacity to replicate. And you can see this here. If you take the protein purified from bacteria, you agitate it a little bit, it will form these long fibers. And if you take these and use them to seed a small amount of into a vial of native protein, you see very interesting behavior. So here's just the native protein alone, and we're going to follow it with an intercalating dye, thioflavin T. You can use some others. And just when you get signal, that tells you started to form a fiber. Okay. And you see a long lag, and then you start to see an assembly. But if you take a small amount of this material and seed a new reaction, you see a totally different behavior, this burst. And that comes from the faithful self-templating, basically this transmission of this structure onto the new monomers of that protein, sort of building fibers. And that's kind of it. That's the basic biochemistry that allows the change in the shape of this protein to confer traits through a lineage in the same way that we would have for a DNA. Okay? Okay, so in fact, it's a little more complicated because that just makes a long fiber that actually that would be stuck in the mother cell. So it is fragmented by chaperones, right, which we think of as helping against protein aggregates. Okay? But in this case, what they are doing is accelerating their growth by breaking the fiber into smaller pieces, kind of seeds, if you will, and then each of those can grow from its ends. Okay? And it's those pieces that are, we think of really as the replicons. They're passed to daughter cells where they have new rounds of growth. Okay? And so just as when we think about replication of the genome being not just one strand of DNA, you need to have the replication cycle and a mitotic apparatus to separate it, all of this to make inheritance work, right, at cell division. The same is true here. You need to break this into smaller pieces. The protein subunits you can think of as the nucleotides in the system. And in a way, the chaperone, again, is making these smaller seeds that are able to be broadcast, almost like the mitotic apparatus in a division of chromosomes. Okay, so because of this, these elements are very strange in terms of their genetics. And this was one of the clues that excited and baffled people early on in their discovery. So these traits are dominant. If you take a strain that, again, these have the same genome, but the protein is in its normal native fold here, and its assembled fold here. You mate them, all of the protein is converted, so the traits are dominant. And then that's stable mitotically, but if you put them through meiosis, the chromosomes segregate, they mix and match, Usually, they should go to half if it's a mutation, half of all progeny. But in this case, it goes to all of them. Okay, so they defy Mendel's laws because they are not attached to a chromosome, right, in the same way. Okay, so when I started thinking about this problem, right, these elements were seen as paradigm shifting but rare. It motivated 2 Nobel Prizes, right? Obviously, people were very excited about the hypothesis, but they were always thought to be kind of toxic for the organism and driven by amyloid conformation. Okay? That seemed like kind of the ground truth that we were taught in school. Okay. But of course, there has been a revolution in cell biology in the last 10 years that has appreciated that cells are amazing material scientists. And they can take proteins and other biomolecules and put them together to form all kinds of non-Newtonian fluids, various gels, all kinds of things. And we began to wonder if some of these types of assemblies might also be capable of holding information and transmitting it. And what we did was to start by just taking a proteome-wide view. And we began in yeast, and subsequently we have done this in many other systems. But the basic idea was to think about the thermodynamics of these folding events that are really unusual. And the assembled state, so the fiber, for example, is actually more thermodynamically stable than the native fold of the protein, which is crazy. But there's a high kinetic energy barrier between them. And so as a result, under normal conditions, you just make the native protein. That's what you keep making, right? However, once you do make the misfolded one or the assembled one, then it acts as its own catalyst. And basically, all the protein is converted, and all future protein is also converted into the assembled state. And that propagates through a lineage. And so our idea was to basically take each protein individually in the yeast proteome and overexpress them one by one so that we would increase the likelihood that you would go into this assembling state. And if you do this, then it should produce daughters and daughters and daughters that are able to keep doing that. And if it gives rise to a trait that we can measure, then we can understand it better. Okay, so we did this. I wouldn't tell you about it if it didn't work, but we took every protein. We did this individually in quadruplicate with small population sizes, so you avoid getting mutations. And then we got rid of the construct we used to overproduce the protein. We go for 100 divisions after the normal, with normal expression. So all of that protein we made initially is dead and gone. It's diluted away. And then we ask, do the progeny behave differently? And indeed, they do in lots of different ways. And we found all kinds of phenotypic diversity in these experiments. Many of them had non-Mendelian patterns of inheritance and all the properties that we associate with a prion-based trait and that distinguish it from genetic mutation. Okay, so we found dozens of these, and they do fascinating things. They are, basically all intrinsically disordered proteins, and most of them don't form amyloid fibers. They instead form these bizarre particles that are often more gel-like, sometimes like a viscous fluid. They can be made in vitro just as well. And those particles that we make there, you can use to transform naive cells just as you would with a piece of DNA. So this to me is kind of like the ultimate proof that that particle holds information, right? That you can make a cell that receives it and its descendants have the same trait. And moreover, we found many specific stimuli that strongly induce these. Okay, so it was, I always thought of these, I think, coming from a mutation background as like a protein folding accident. Okay? That maybe sometimes it can be good, often it's bad, etc. But again, many of these are present naturally in a population at, let's say, 1 in 10,000 cells, 100,000 cells. But in response to a stimulus, they become present in all cells. Okay? So it's almost in that way like a stress response. Okay? And in extreme cases, actually, the particle itself, its characteristics, its size, the way in which it's organized, etc., actually differs depending upon the amount of the signal you add. And this appears to provide a way in which actually the detail of the stimulus can be memorized for multiple generations following the production of the particle. All right. So here is an example that is coming from an RNA binding protein that actually is, again, deeply conserved from us to down to yeast. And in us, and in fact in yeast it does something similar, it is super important in making sure that you make your own transcriptome. So for the first couple of cell cycles after fertilization, basically you wind up using mRNAs that mom gave you. And then you need to destroy them, okay, so that you will give way for your own transcriptome to be produced from your own genome. And this is the protein that orchestrates that process. Okay? So it's called SMOG in Drosophila, SAMD4 A and B in us, and VTS1 in yeast, but these details don't much matter. What we discovered was that what we think of as the native fold of this protein can bind to about 200 mRNAs and direct their degradation. The assembled form of this protein, right, can bind all of those and 100 new ones as well. Okay. And that is because it is using the kind of preformed avidity that you get from being assembled in these structures to bind better, just as a tighter KD, but also actually the downstream effectors that actually do the degradation itself are recruited into these particles incredibly efficiently, much more so than they are capable of forming a one-to-one complex with the diffuse protein. And it turns out that these targets aren't random. They are orchestrating a trait. So they wind up encoding proteins that regulate a key decision about whether you're going to take energy you have and turn it into more proliferation, or instead, in this case, differentiate through meiosis into a spore. Okay? And this assembled state favors very robust proliferation. And interestingly, in cancers, this protein can be overexpressed by 50-fold to get into a state that it is actually assembling in a great deal. Okay, so we found many other examples. They do a lot of cool things. You can read about it. I don't go into great detail. But really controlling pretty fundamental aspects of biological regulation and cell biology. And I think an important point to emphasize here is that they are hidden in plain sight, right? Like we only know that the red cells and white cells are different because someone put artificially a stop codon in this adenine biosynthetic gene. Otherwise, they look identical, right? And in fact, I will say that this example I showed you before, it turns out that the form that looks like this, that is what we think of as the wild type native form in anyone who has done an experiment in yeast. You have worked with that unknowingly, right? And that is because the history of laboratory husbandry, as you are growing and growing and growing, you're passively selecting, right, for more proliferation, right? And again, you would never know, right? There are thousands of papers about growth in yeast, right? No one would know that this was there unless you knew to look for it. Okay. And you can see wild differences in cell biology. There are examples that go the opposite way, where you have dots that are formed in the naive and not in the prion plus, et cetera. And other cases just beyond belief. Okay. So we've wondered kind of how widespread is this behavior? Is this just kind of an oddity of yeast? They have a very AT-rich genome. There's a lot of asparagines and glutamines that we tend to think of as driving this behavior. Or might it be more common? And we started to think about what kind of a trait would be, again, universal to these. Not just do they grow better in an antibiotic, but would every single one of these transitions have? And we settled on the thing that Prusiner himself first found, which was that the assembled protein is resistant to a protease a little bit. Okay? And the degree can vary depending upon the protein, but it is a general property and something that we could probably assay. So here are those data, and what he saw were these plaques, but he found that if you just first do the lysate and don't add any protease inhibitors, you see some degradation. And then if you add protein over time, or protease, you see that there's degradation over time, and this species remained, which is from the prion protein. And here's with the protease. Okay, so we thought, what can you do that would allow us to do this again, proteome-wide and in cells? And we started experimenting with different degrons, so these tags that allow you to control the degradation of specific proteins. And we settle on one that is, rather than being inducible, it's actually ligand stabilizable. So it's kind of on all the time, and then you can make it a little bit less on by adding stabilizers. And spent a lot of time playing with these sequences between. So between the open reading frame and the degron, and then a marker at the other end. These are directional degrons, so they start degrading from here. And the idea was basically, if you assemble, can you then block the capacity of the proteasome to keep chewing through the protein, and thus preserve the activity of the marker that's on the other side. Okay. And then we decided to try to use yeast as kind of a test tube at the beginning, mostly because it's so easy to manipulate them, and because you can go through division after division after division without any concern. And so you don't even have to make any assumption about the half-life of the protein you're using. You can just go through so much division that you would dilute it more than 1 billion-fold, more than 10 billion-fold, so there isn't any molecule left that was there at the beginning. Okay. And so then we tried to use this reporter with a known prion. So this is looking at the prion domain of a yeast prion called MOT3. And then a version of the protein where we remove the sequence that's important to make it into a prion. And then we just ask, can we select for a marker? So in this case, we're using a nutritional marker. And what you see is that initially, the ratio is close to 50-50. And then as you start selecting and selecting through repeated rounds, you enrich it, just like you would a mutant, except, again, there's no mutation here. You can sequence it. And it is just fine. That comes from the protection of the marker at the N-terminus from the assembly of the protein. Okay. So ultimately, I'm always worried in these genetic screens if you find something weird, right? It's not what you intended to find. And so as a test, we asked if we take the winners here and we now cross them to a cell in which we tag the protein with GFP, did they switch? And indeed, they do. So these are the ones that we didn't do anything to, and they have MOT3 in the nucleus as the transcription factor it's supposed to be. And here are the ones that switched, and what you see are these cytoplasmic foci, just as you would get if you made MOT3 in a directed way instead. Okay. So we then moved on to test the human orpheome. So this is about 15,000 clones that have been assembled by Mark Vidal and others at Dana-Farber over the years. They cover a large fraction of the human proteome. And using gateway cloning, we put it into this construct. And then in multiple flasks, we just do the selections. It sounds crazy, but the whole process is under 2 months to finish. And basically, you just sequence your way out of it, right? So you ask, what's the composition of my pool at the beginning? And then at each one of these steps, and you look for each clone. Is it enriched? Is it depleted? And give it a score. And you can learn a lot of things about the switching, but also toxicity, et cetera. And if you look at the winners, you see this fascinating result. So this is FTO, which is a protein that removes M6A modifications on RNA. And here you see kind of a temporary enrichment, right? So initially it's growing into the population, and then eventually it's depleted. This protein forms in vitro a condensate that doesn't have the capacity to propagate very extensively. A little bit, but it's actually relatively weak. By contrast, here, MRPL10, which I should probably call UL15M, I think it is, if I use the formal nomenclature. It's a mitochondrial ribosomal protein, and it is enriched dramatically, just as if it would be a mutant, and actually stronger than the control prion that I showed you before. Okay. So the beauty of doing this outside of the native organism is that it's relatively easy to ask if it's from the protein. So you can just stop expressing the protein for a couple of generations and then express it again, right? And if it's a mutation, it should come back, right? If it is from this mechanism, you didn't have the nucleotides of the system, right? You weren't making the protein. And as a result, you should eliminate the trait. And indeed, what we found, that is in fact what we found. So the majority of these require the continuous expression of the protein, as you would predict if it is from this kind of assembly process. The purified proteins do crazy things. So here's that FTO. It forms roundish objects that have recovery times that look like condensates. They fuse. That's all normal. Many of the others, they don't form fibers, but they form these bizarre extended structures that are very interesting. We understand much less about them, but nonetheless, if you take them, you can use them to transform naive cells. Okay. And indeed, you can do this also with mammalian cell culture. Many people believe that disordered sequences are the only way to do this. And I don't know that I believe that. So we started to look at other proteomes that we were told can't do this. So we looked at the E. coli proteome, for example, that has many, many fewer N's and Q's, and tried to ask, would it have maybe less of this behavior? Not at all. It was, in fact, equally abundant, if not even more so. The biases you uncover are different, but the traits that can be elicited by these elements are huge. So this comes from the ancestral transient overexpression of FTSY, and this assay we're just looking at the swimming of the E. coli over time in the soft agar. And as you can see, the ones that were derived from that population are much, much better able to swim. And that's just one example. There's many cases of antibiotic resistance, other traits that are fascinating, and in fact, as large or larger than genetic mutants that we know to affect the same phenotypes. Okay, so in the human proteome, we see very interesting behaviors with respect to charge distribution in many of the proteins that are winners. And often the proteins themselves are not especially positively or negatively charged, but you have a region in which it is extremely biased that turns out to coincide with what is required for the behavior. So just some of the more extreme examples. But there are other kinds of patterns as well that we're trying to understand more deeply. And the enrichments that you see, some of them make a lot of sense, right? They are enriched for proteins that are binding to themselves. And in fact, in almost all the cases we have looked at deeply, dimerization is an obligate first step to engaging in the rest of the assembly process. And they occur in all kinds of, they tend to be attached to other domains that we have found before. So sequences like those that are involved in that RNA binding protein I showed you before, but also many others. And interesting places where they are. So I think it doesn't surprise anyone that we would find them in proteins that are localized to the central nervous system. But we also see strong enrichments, in fact, stronger enrichments for the reproductive system, as well as for the GI tract, which I think are, and you'll see more about that in a moment. Also, in the nucleolus and membraneless organelles, those things shouldn't surprise us. But strong enrichment for localization to exosomes and other kind of vesicles that are actually thrown out from cells. People often think of this as sending trash. I think it's provocative to think about it as potentially a signaling mechanism. And indeed, if you look at enriched functions, we see roles in stress responses, also post-transcriptional gene regulation, like I just showed you, but also differentiation early in development and cell cycle. There are a lot of proteins that are involved in progression at G2M that seem to have this property. And finally, protein quality control, and I think most interestingly, it functions in immunity and in communication. And I think, as an information storage device, that type of behavior may make a lot of sense. All right. We then, this library included a lot of clones that initially annoyed me because it's, you'd have the wild type copy of the gene and then some other things. Mutants, right? I'm like, why is this there? You're wasting real estate that you could have used to incorporate more genes, right? But it turned out they are not naively put there. They were put there because they're interesting comparisons between the wild type and a common disease allele, or a common variant in human populations and a rare variant, et cetera. Okay? And it turns out that many, many disease mutations alter this behavior. And in fact, the alteration of the behavior in this construct is a better predictor of pathogenicity among all Mendelian disease variants we've looked at than any other metric that you can make computationally, for example. So I think it's, it tells you that stability of a protein and its assembly status matter a great deal. But some of these were really remarkable. So this is a cyclin D mutant. So the wild type is degraded as you would expect in this assay. But this is a mutant that's associated with a couple of cancers, right? And yeah, it clearly assembles, and you can see this in the cell biology as well. Very striking. Okay? And again, this is just what I was telling you before, that in fact, if you look here at in non-disease variants in the same proteins, you find that they very rarely affect the assembly property, whereas the disease variants very commonly do so. And it's not that you gain the capacity or you lose it, or any of it can happen. Just the change is associated with disease. Okay. And again, this is actually a better predictor than even the sum of all algorithms that are currently used in clinical genetics. But then comes the surprise. So the fact that there was an enrichment in factors that are involved in communication between cells made us wonder about effects that might extend beyond the organism. And we started to see strange behaviors. So a dogma in the prion field is that there are strong species barriers that affect transmission from one organism to another. So most of us who ate beef during the period where we were not scanning for mad cow disease didn't get sick, right? And in part that's because the transmission of aggregation from human to human is very efficient, from bovine to bovine, very efficient. From the bovine to human is 10,000-fold less efficient. Okay? And these didn't have it. So, for example, the protein the smog I showed you before, you can look at its capacity to be transmitted onto a homologous protein from an organism 15 million years separated. It's fine, it's totally capable of doing that. Okay? But even if you look for 1 billion years to S. pombi, still totally fine, right? That protein is really substantially diverged. And it's not that this will just make any protein aggregate, it's selected for this protein. Right? And this made us wonder about kind of more complex systems and whether it might be possible to transmit information this way, more broadly. Okay. We also had seen contemporaneously that actually interaction with the bacteria in the environment could lead to, again, uniform induction of a prion that we called GAR. And that this, in fact, again, the strongest known inducer is secreted by a competitor. And of course, we are covered in microbes, including fungi. And we don't usually think about this. People just sequence 16S. But it turns out that a couple % of our microbiome is indeed from Cerevisiae and even other fungi as well. And in nature, this organism that we use as a kind of workhorse in the laboratory turns out to be a major component of the microbiome of insects, in particular flies and also bees and wasps. And so we wondered if these elements that, again, seem to be incredibly conserved, right, might have a role there that would extend beyond the interesting but frankly often modest changes we saw in their growth in different stress conditions. So we began to ask about this, and we turned to Drosophila in collaboration with Andy Clark, who ambushed me at a meeting about this, and this turned into a great collaboration. So he is at Cornell, and Chichan Lin, his postdoc, helped us out with this. But basically the idea is to take Drosophila that have been synchronized, they're all females, and then they are germ-free, and then to introduce these yeasts to them. Okay? One by one, there are 40 different ones that we try, and they have, again, the same genome, but in each one of them, an individual protein is in this assembled state that is heritable, and then we have controls that are totally naive. And then we ask, as adults, as we keep feeding them, what happens to their behaviors and lifespan, et cetera. And many, it turned out, had strong effects. Okay? So it was really surprising to me how many of them did. So these are some examples. We see plenty that give rise to heat resistance, improved climbing performance, and I will show you in detail for a different one. But this is startling. It's I have to say they are massive phenotypes compared to what we see for the yeast themselves. So ordinarily, if you take flies and you put them at 4 degrees, they will go into what is called a chill coma. They stop moving, and then you can heat them up, and they will come back to animation. They keep going, and you can do this through many cycles. But if you feed yeast that have this protein that has been assembled. This is a mitochondrial ribosomal protein, the same one I showed you earlier in the screen. They don't do it. They just are still moving around. And there's no apparent cost to them. You can keep doing this over and over again, and they are just fine. They just don't go into the coma. Okay. And this effect turns out to be almost homeopathic. So you can dilute and dilute. So you give them smaller fractions of their diet are this one that have the prion, and others of them don't. So even down to, in this case, 1 in 200 cells, and you're still able to get a very strong response. Okay? And this tells you the signal is very potent, it's durable, and in fact it's even stable if you will chase it with naive yeast. So they only get their first feeding is this one, and then you keep giving them only the naive afterward. Okay? And you can microgavage them with heat-killed fungi, the same effect. And interestingly, there is a preference. So once they eat this one, they want it in the future. After it's almost like a mind control by the yeast. Okay, and it is not just about cold tolerance. So these flies actually just move more in general. They're more active. And in particular, in the night, and these dark cycles where they're supposed to be resting more, they're far more active. Although in this kind of these siestas in the day, maybe not so much, right? It's kind of interesting. Okay, so what is going on? We still don't totally know. But we started to use these global diversity lines, these accessions of Drosophila from different places, and do basically a genome-wide association study on the response to this element. And then we take also diverse libraries of yeast that have been isolated from patients, from grapes, from the bark of trees, from soil, all kinds of different environments. And then ask if we can understand, through all of this, what types of genes regulate it, and anything about the ecology. And the beauty of Drosophila, well, there are many beauties, but one of them is that the RNAi is actually pretty easy. So you can do the pretty good validation of the hits you find. So in doing this, we found about 67 genes that had an effect. It's important to remember the gene needs to be polymorphic here. And basically, they can trace a track from the gut to the brain that appear to be involved in this behavior. So these are all genes that when you knock them down, it either goes away or it is enhanced. And here you can see the same thing about where they are localized, that kind of map this axis from the Malpighian tubules through to the central nervous system. So we are still trying to figure this out. We have some indications that at the end there is a competition between serotonin and octopamine signaling that is involved in the trait. However, the ecology, I think, is one of the most fascinating things of this, right? So like, why go through all of this, right? And one of the ideas we have is that actually this leads to an ecological benefit. You get better dispersal for the yeast. And in fact, if you look at fungi that are derived from cold ecological niches, they are enriched in having this aggregate naturally. Okay, so now as time gets short, I just try to finish in a couple of minutes. But this all seems great, right? So I've told you about how the collective behavior of biomolecules can let them do things that no individual one of them could do alone, right? And yet we also know that that aggregation is something we think of as being bad, obviously. And of course, it is linked to all kinds of devastating neurological diseases. And so, can these two competing facts be reconciled? Is it just that different proteins are different, or is there some yin and yang to this? And to do this, we used a kind of supermodel of aging that is a fish called the African turquoise killifish. It is the shortest lived vertebrate that can be bred in captivity. And in about 4 to 6 months, depending on how you grow them, it goes from birth to death. But what is amazing is that all of these transitions, right, that we go through in decades, it's going through in the same fraction of its lifespan here. So this model has been exciting to ecologists for a long time, but there were no genetics, and my colleague Anne Brunet changed that a couple of years ago, and it became a very interesting model to start using. It gets cancer, cataracts, has learning deficits, all the things that would happen to us as we get older, right? And Yuan Chen in my lab basically developed a way to very carefully isolate aggregates in young and old animals across many different tissues and figure out what the proteome looked like basically through the course of aging. And after quite a lot of experiment, she discovered many proteins that aggregate, and they do it in the context of natural aging. I think what is very exciting is how many of them overlapped with the results from the screen we did of the human proteome for the self-assembly behavior, okay? And so, in fact, with some of them we follow up, but it suggests that, like, perhaps some of the spread of aging throughout a tissue comes from this kind of transmission behavior. And many of those condensates or aggregates I showed you before, if you wait and wait and wait, they will start to form a much more deranged structure, something that many chemical engineers would call an arrested condensate that can cause a problem. Okay. And in fact, as a final test of this, we ask back again in yeast, right? If this is true, then I should see an effect on aging directly. So how to frame this? You can look here in this microfluidic chip, as you keep dividing and dividing. These are mothers, and they're producing daughters. And you can count the age, basically, by how many times they divide. Okay? And then she'll sit there, and then eventually there'll be a metabolic collapse, right? And people say when you stop dividing, that's death, but they're not dead. So now that I think I'm done having children, I'm very interested in this, like the post-division life. But in any case, so you can use this to measure their life. And the question we had is, if you get rid of preonic elements that these yeast have, what happens to them, right? So unambiguously, for young cells, it's better to have them. It gives them all kinds of advantages in stress. Okay? In fact, it's kind of a weird thing in lifespan. This is fascinating. But like every variation, every mutant that either makes lifespan longer or even shorter, they actually have exactly the same shape. They are just kind of moved one way or the other way. But if you divide them by the midpoint, they all scale to the same thing. Okay. So this means that like both health span and sick span are extended. So if you live longer, it's great to live longer, but also this like trail is terrible. So you have a long period of time in which you're not doing very well. Okay, so we've developed some models. Again, we don't need to talk about this in great detail. But the thing that was very exciting is that if you remove these prion elements, again, in youth, you stress them, it's much worse. You don't want to get rid of them. But in aging, it's great. These cells live a lot longer than if they had those elements. And they also, when you scale them, don't have the same shape. They make this very steep, almost I call it like a democratized death. Everyone lives a long time, and then collectively they're done. And that turns out to be exactly what you expect from rare interventions that actually will enhance health span and then not have that same effect on sick span, basically limit it. And this, I think, raises interesting questions about whether that kind of conformational conversion is an important part of the aging process that we want to investigate more in the future. Okay. And indeed, we examine every survival curve that has been done in yeast, and this turns out to be really the core of what is responsible for this type of aging. Okay. So take-homes, non-genetic mechanisms are everywhere. They're hidden in plain sight. You can't sequence your way out of them unless you know what to do, but it doesn't mean that they are not important. And in particular, the capacity to assemble into different collective forms allows biomolecules to do things that none of them would be able to do individually, and can even can transfer information across generations. I think the stress responses clearly can promote this behavior. And also, it reveals different targets that might be interesting to explore. So, there are specific chaperones, for example, that are involved in the fragmentation of the larger objects to create these seeds that transmit from one generation to the next. And you can inhibit those easily. There are some that are only present in microbes, for example, and they might be very interesting targets as antibiotics or to prevent the acquisition of resistance to them. Okay. And again, these diverse strategies inform each other, right? The transient response can induce more mutagenesis, for example, to make the more permanent one, and vice versa. I just show you here a case of isolates from patients. This is from someone at Mass General Hospital who has HIV or had HIV. And it's an assay for mutation. And what we find is that many of these isolates have acquired a prion-based mechanism for diversifying, right, for basically becoming more resistant themselves. Okay. So to close, this kind of resilience, in fact, I think comes from the adaptability. And we are used to kind of gazing at genomes, right? And it's incredibly powerful, but it's sort of the fossil record. There's snapshots in time that we have. And really, I think one of the great challenges for us going forward is understanding the much more dynamic picture that was involved in getting us to those snapshots and how we can control this. And I think we are in a golden time, both experimentally, but also in terms of AI, other types of approaches to try to get a better understanding of those intervening steps. Okay. And with that, I thank you for your attention, as well as those who did the work. I talked about things that basically everyone on this screen did, along with these collaborators without whom it would have been possible or even as fun, the kind folks who funded it, and you for your attention. I'd be very happy to take any questions. Thank you.
Thank you very much for so fantastic talk. So, preguntas, questions? No, questions? A ver, allá arriba. ¿Vas corriendo? Y luego, ¿quién alzó la mano? Enrique, tú. ¿Ahora estás la mano tú? No. ¿Alguien más la alzó? No.
Hi again. Thank you for the seminar. I really liked it. So my questions are regarding the first part that you talked about, well, mainly biochemistry. Mm-hmm. So when you said that these prions can assemble in like gels within the cell, I remember like the phase-phase separation thing. And I wanted to ask if there is like some relation with them, if these prions can trap other molecules and then promote this phase-phase separation, or what's?
That's a great question. So I will say when we try to like mutate individual proteins to death, it's very hard to find mutations that will block the phase separation but not the prionic behavior, and vice versa. So even if they are not exactly identical, I would favor the idea that like one is on pathway to the other. The gels are fascinating because of course they have pores, right, that are also very big. And this chemical engineer in my lab who has this kind of crazy model that in fact that like selective recruitment of the executioners for this RNA degradation is about basically those forming a large-ish assembly that's able to fit inside of the pores. I think our kind of intuition for like what structure and reactivity is like is all based on kind of like angstrom scales of interacting of a protein with a small molecule, this kind of thing. These are on a much larger length scale, and it's sort of hard to know how to think about it. But indeed, there is a surface of those pores that is formed, and at least in simulations, it has interesting properties that would probably enable selective binding. So.
Okay, and also I wanted to ask if those 30% of the proteome that you said that it's not order, are they potential prions?
Like how many? Yeah. Okay, so now I didn't include this because it's like I find it like wild, but I will tell you now. So if we look at the human orpheome in this assay, 26% of the orphs are having some degree of propagation that goes beyond what you should expect, right? Of assembly beyond what you expect, rather. And then of those, 2/3 of them have some degree of propagation that goes beyond what you should expect, right? It's a lot. If you would take the kind of state-of-the-art algorithms to predict prionic behavior, you'd pick 0.1% of the proteome would be high enough. Now, a lot of that is not as stable very, very long term as some of the examples I showed you. It's more for, let's say, 25 divisions, right? But that's kind of already plenty long. And I think this is one challenge in this field that people have almost had a fundamentalism about what is a prion. it needs to be a particle that is really infectious from like one individual to another in this way. And it has kind of closed us off to actually many other exciting examples where the same type of biochemistry is restricted to transmission, let's say, within a tissue, right? Or even that allows memory to persist within a cell that experienced a stimulus for an entire lifetime, right? But that is spatially sequestered from passing to daughters, for example. And so I think that behavior, certainly the data suggests that it is very common. We've looked at now probably 36 of these in vitro. It all looks very similar. And so, yes, many proteins appear to at least alone, right, have a capacity to, in a protein autonomous way, store and transmit information.
Thank you.
Hey, Dan, that was a great talk. I have 2 questions. One of them is based on the shared properties of these cell template proteins. The one that I would like to ask you if you could maybe speculate or you maybe know a little bit more of why is this charge patterning? Why this particular charge patterning would have an advantage for these proteins to perform this function?
Yeah, I mean, this is also a great question, and of course it's impossible to recode everything such that it's at that point no longer the same protein, right, to answer. I think many of them, they are interacting with other very charged molecules, and they often, if you mix them together, it has a large effect on the assembly property. And so it may be that this is part of it, right, that it's kind of an interaction with self versus interaction with another partner. For what it's worth, the domains seem to be sufficient, so you can attach them to GFP and get them to behave also as a prion. So it cannot be all about self-self interaction. There are other behaviors that we see arising, many kind of like, we're just thinking about stickers and spacers in this field, but we see many examples where the stickers, these are like small motifs within, let's say, a disordered region, that basically give you multivalency. And the kind of prevailing idea in the context of sort of garden variety phase separation is that they are isoenergetic interactions. And these, actually, they are not. There are like a couple of them that are really strong, or at least you would predict to be strong, and those interactions are extremely important for guiding the assembly process down one that is going to be capable of propagation. And you can break, you can swap, all this kind of stuff. So yeah, there is something there. I think it requires a lot more to really fully understand. And of course, the risk of all of this kind of stuff, we train AIs on it and this kind of thing, but there's so many sequences and probably different biochemical origins of the behavior that it winds up obscuring the signal from any one. So I think this for the future, kind of even classifying them is an important step to understanding more.
Yeah, because maybe thinking on making a synthetic protein that has these features and testing whether that can act as a prion.
Would it be sufficient? Indeed, it's a beautiful test, yeah. And I think the other thing that is very exciting to me are these disease variants, right? That kind of that all of the predictive power we have now looks over sequence biases in large domains, right? And so if you would make a single mutation, the prediction is the same. It should be capable of having the same behavior. And yet lots of them that are involved in disease don't, right? And I think trying to tease apart like why it is that specific mutation blocks the assembly is a very interesting way forward to kind of dig deeper into what the interactions are that matter. Yeah, that's great.
I think we have a question there for Zoom. Can you read it?
Yeah, so it says, regarding the C. elegans aging-related experiments, if we overexpress, maybe via some more active promoters, the proteases responsible of aggregate degradation, do C. elegans emulate these cured organisms?
Would you help? Yeah, it's a great question, right? So if you could like have the aggregate eraser, for example, right, would that be beneficial? I mean, I think it is unknown in the context of normal aging, and that's sort of why the experiment I showed was exciting to me. Certainly for disease, it has not been very helpful. Right? There's been a lot of effort to try to kind of make an antibody or other molecule that will get rid of an aggregate, and they have not, in general, been fantastic therapeutics. And in fact, there's a lot of evidence that the sort of forming the aggregate is part of a protective mechanism or an attempt by neurons and other cells to avoid death, right? So it is, of course, correlated with having a pathology, but not perhaps what does you in. In development, right, we know we need these types of assemblies in many cases. In innate immunity, we know that we need this kind of behavior in various inflammasome, in the MAVs, for example, assembly. And so probably you would not want to always get rid of it, but I think it's an interesting question of if you could do it selectively at a specific time, what would happen? And this awaits to be seen.
You just made me think about a weird experiment. Thinking about these big, great complexes that have these big pores and then make things interact with each other. So it's probably very difficult just to imagine how to explore that. But I was thinking that you can probably express one of these prion-like or aggregating proteins and bind it to something like a paramagnetic bead, and then let it interact with a whole cell protein extract and probably add a little more of the protein in solution, and then pull it away. Are you aware of that kind of experiment?
We have even done that. This is so crazy that we did. I did not talk about it because it is too complicated for people to digest it in a one seminar. But we think of this type of behavior as being totally homotypic, right? And that it just involves interaction of one protein with itself. And clearly, that is sufficient to give rise to the behavior. But indeed, in actual living cells, they aren't just floating around alone. They interact with partners. But what is crazy is that they are really reshaping kind of the proteins that were often part of the same complex to begin with. And now they are interacting in a different way. And so we looked at, for example, histone deacetylase complex, a couple of subunits that would be there in the normal complex, right? In the assembled form, they're not there at all, right? One of the other ones, its stoichiometry is increased twofold. There are some new interactors, right? And it's not that every protein is going into this blob. It's still very selective, but it is a very interesting logic. And if you look at kind of what that means in terms of regulation, it gives rise to very exciting outcomes. So I think the behavior of this is more sophisticated than just simple switching of one molecule. And indeed, experiments like that offer important ways forward to understand better.
So this is a question on Zoom. So it says, back to the example of MRPL10 effect on Drosophila. What do you think MRPL10 is doing inside the fly? Is it causing some endogenous?
Yeah, exactly. Yeah.
So is it causing some endogenous protein to
Oh, it's fine. You have a loud voice. It's fine. They cannot hear. Okay.
Oh yeah. So here we go again. So back to the example of MRPL10 effect on Drosophila. What do you think MRPL10 is doing inside the fly? Is it causing some endogenous protein to adopt a prion structure? If so, would that be inherited in the flies after exposure to the exogenous MRPL10?
That's an excellent question. So I initially thought that idea was heretical. I thought it was, it is a mitochondrial ribosomal protein. Maybe it is shifting its assembly status, and then the translation in the ribosome is different. There's some difference in respiration and the metabolic change, right? That would be responsible for the effect. In practice, this kind of dilution experiment I showed you suggests that probably that is not it. We've also tried to use extractions from lysates of cells that will allow you to get out small molecules and then resuspend them and then put them back in. And this doesn't lead to the transmission of the trait. And so it led us to start thinking more about the actual propagation of the protein. And indeed, we know both in the cell-based assays and also in vitro that the assembly of the yeast protein is able to seed the Drosophila protein, the equivalent, which is confusingly called MRPL15. So that's why I say the UL15M. But in any case, so you can definitely efficiently cross-template in this case. The imaging in Drosophila is harder, and we are not really there yet for me to say confidently that it's very clear that is happening in the gut epithelium, but I think it's at least an intriguing hypothesis. We've looked at some sort of selective mutants of serotonin and octopamine receptors in specific neurons. And there you can definitely have some that will either block the effect or make it happen even in the absence of the transmission. And so there's definitely, by the time you get to the neuron, something like this, we don't know whether the protein itself travels. I think for alpha-synuclein in Parkinson's, there is also a hypothesis that initially I thought was really crazy, but starts to become more and more interesting over time. And basically, patients who have some types of surgeries that block interaction between the gut and the brain have very little what we would call like a gut-first Parkinsonism. And in fact, people who have gut-first Parkinsonism have an increased amount of E. coli in their microbiomes that express amyloid at its surface known as curly. This is how it builds biofilms. And you can see that curly can very efficiently cross-seed alpha-synuclein, and there's some kind of suggestive, maybe not completely conclusive, but suggestive studies in mice suggesting that you can, in fact, induce pathology in the brain by having a microbiome that has this, by introducing the curly, et cetera. And it is at least an interesting hypothesis to consider that something similar may be happening here in a way that doesn't have an obvious detriment to the fly, at least in the conditions we look at. I think it would be interesting to look in all those experiments in the mice, for example, or in a genetic background that is sensitized, right, to develop Parkinsonism or synucleinopathy. And I think it would be interesting to look for these as well in those types of backgrounds to see if there is, again, some cost, if there is a particular genetic sensitivity to the disease.
Hello. I was just wondering, is there any effect on the formation of prions by post-reductional modifications?
Yes, that's a great question. So those sequences that are enriched in these screens are in fact also enriched and being modified in various ways. So the two of them that are the most enriched and in fact in our hands seem to have the greatest effect on the switching behavior are phosphorylation and then arginine methylation. Both matter a great deal. And in fact, those modifications, very often bracketing short sequences that appear to be nucleation motifs, wind up being like major control elements. So for some prions, we have seen indeed that like one or two phosphorylation sites are enough to lead to conversion or loss. There's an interesting coincidence for many of them with sites of 1433 binding that is specific to phosphorylation of the motif. And it makes me wonder a little bit about whether there is chaperone recruitment that is important for fragmentation or what is happening there. But the answer is very clearly yes. And in fact, if we want to make a variant that is either obligate prion on or obligate prion off, the first thing that we do is look at suspected phosphosites and make phosphomimic and phosphodead, and very often you get a winner.
Yes, so how do intracellular conditions like calcium, intracellular concentration, and pH affect or alter prion formation or change conformation?
That's great. So in vitro, we know that indeed those kind of changes can have a large effect on some of the proteins. Interestingly, again, many of them have a isoelectric point that is derived in a very unbalanced way from different parts of the molecule, and I think that may be, again, a little bit of what you're getting from this weird charge patterning. In cells is also a good question. Repeated, so in some cases we know a stimulus that just like one stimulus is enough, right? In other cases, like a repeated amount of you kind of hit it and you hit it and you hit it again is a better inducer, right? The reason for this we don't 100% know, but it may be you need to kind of keep sampling the conformation. Again, the detail of whether this comes from direct binding, I think we don't yet have such an example, although I think it's an interesting model to consider. We have lots of examples of a post-translational modification that came as a result of that kind of stimulus.
Thank you. Thank you, Dan. I have a couple of questions about the prions in aging. I was wondering if does self-template behavior is an accumulative process, or is there one trigger in aging, and if that in case that one trigger was responsible for that behavior, is there a genetic cause, or it's inherited by these mechanisms through prions?
That's a great question. The fascinating topic, and one that I always wonder. So, in the fish, in the aggregate, you see protein that have a prion-like quality. You also see chaperones that act on those specific types of protein, right? So, like in the brain of the old fish, in fact, these glutamine and asparagine-rich proteins aggregate to a ridiculous degree. And specific HSP40s that respond to those, but not to other types of proteins, right, are co-aggregating with it. And it leads to this kind of chicken or egg question, right? Is it that the aggregation happened first, right, and then there was sort of the chaperone demise? And in fact, again, as you aggregate, you're depleting the chaperone from the soluble pool, and basically you wind up having this kind of self-amplifying cycle that gets worse and worse over time. It doesn't, it kind of changes the goalposts, right? Like what would be responsible for first the chaperone decline, right? So there's some nice studies in worms from Rick Morimoto and some other organisms too that suggest it isn't just that like we get old and experience more stress, but that actually in many organisms at the point of reproduction, there is actually like a programmed reduction in the amount of chaperone that is made. And that just stays. It just doesn't come back, right? And I think it's an interesting question of like how much of it is really damage versus like in a way a form of development, right? That you're kind of like done becoming who you're going to be, and it's time to invest that energy into the next generation. I think in terms of what causes like the seed, right? Is it just that like eventually one happens? Sure, but then if that's true, perhaps you would expect to have a very similar, risk. You see increased risk with age until you get to be about 80, and then it doesn't matter. You have the same risk after that. So if you make it there, you're in good shape. You stop aging. And there have been many hypotheses. I think one of my favorite ideas is actually errors in translation, which we talked about a little bit yesterday, but they are actually really common. And many of them, we know, can actually seed the assembly of some of these proteins. And there are new mass spec approaches that have been developed to measure this. If you take the proteins from the fish that aggregate and look at them, they in fact have accumulated at low percentages, but a measurable amount, the mistranslation of them to a form that we know is a better seed, basically. So I think this is a intriguing hypothesis. If you take, strangely enough, there are other domains of life. So eubacteria, for example, have a version of RPS23 in the ribosome that is more faithful. And if you make the mutation basically that allows them to make fewer mistakes, you extend lifespan in every organism in which it has been tried, in worms, flies, yeast. No one has tried in humans yet, or in mice. But I think it's an interesting hypothesis that this may be at the core of the problem.
So here they ask whether you could find a relationship between the aggregates in the microbiota and human phenotype. So I guess this is.
That's a great question too. I don't know. Actually, I think it's interesting to ask, even to identify what they might be, like what actually survives the gut, right? And tons of proteases there. It's almost like a version of the limited proteolysis experiment. That exists for human microbiome work, but no one has separated the aggregated or assembled proteome from the rest of it.
Hey, thanks for the seminar. Maybe it's a dumb question, but I was left with the doubt, so I'm gonna ask it. But I don't understand how exactly does the immune system respond to these prions or strange weights or folding. What happened in the organism with these systems and interactions?
No, that's a great question, actually. There are no stupid questions. But this one is actually very good. I think, well, first I will say, like, within the organism itself, right, for even aggregate made by itself, right, it varies dramatically. So, some of them elicit very strong stress response, others of them none at all, right? And I think it speaks to how naive our understanding of what an aggregation really is, right? It's clearly it can have wildly different response for the cell. Some of them can be, toxic in and of themselves, but also they each have individual functions. You lose some of that activity that probably also matters. In terms of immune response, in this context, we understand much, much less. And I was talking with Enrique earlier about kind of how we might rule out hypotheses that this is some kind of immune priming. We know more about that, right, for specific microbes. So Candida albicans, for example, on us, right, is involved in basically making our immune system better until it, if you become immunocompromised, will infect you. And there are very interesting aggregation processes involved in many of the switches that control this behavior, but no one has really looked at the response, of the immune system to the aggregate per se. I think it's an intriguing hypothesis to look at in the future.
So the last from Zoom, I think.
David Gregorio asked, is there any relationship between enhancer activity or chromatin organization and aggregates accumulation?
I think yes. So that's a great question. How do you think about this? So a couple of lines of evidence would suggest this, but the first of them is if you look in, you take various techniques that allow you to ask how kind of closed chromatin is. Let's say you do a TAC-seq and you do this kind of in a limiting way, or you liberate transposons in other ways. What you will see is that there is a kind of a periodicity, so you can see kind of TADs, but also kind of smaller periods within them. And actually, in many of these prion states, those have changed, right? And they are prions that are formed by proteins that we know are involved in chromatin modification or in interaction with chromatin either as a general or specific transcription factor. In terms of enhancers per se, also is a very interesting question. Most of the work we've done has been so far in yeast, and there are enhancers. They are sort of a smaller range, and definitely you can see sort of more activity of some of them in the converted forms of specific TFs. We've done some work in cell culture, human cell culture, with a couple of transcription factors that have very interesting assembly properties. There, you're focused less on the enhancers, but on like their function as pioneer factors, particularly for like FOX transcription factors. And it does appear the capacity, like they stay, as you go into mitosis, they stay stuck on the chromatin for a very long time, almost like they are not coming off. Eventually this happens. But that capacity does depend on, looks like it depends on this capacity to assemble as well. So mutants, at least, that block that behavior leave way earlier and have phenotypes suggesting that they've lost that kind of pioneer activity to the extent someone believes in it. But, yeah.
Okay, so Tere.
Thank you, great talk. I have a question. In the adaptive process of the prion form, the context of the cytoplasm, the different generation are changed, or is only the form of prionic that is hereditary?
That's great question. It is the same. That's like, as we do. The idea is that you are, in theory, expressing the same proteins, right? We did an experiment that I have still not written up, but basically where we use this chaperone curing experiment, and then asked if you kind of do what's called structural proteomics. You do some proteolysis on the whole proteome and watch over time the degradation of specific proteins. And you just say, is there like a difference in the protection pattern? And for at least 500 proteins, there is one that is very clear. And it's very interesting. It suggests that it raises a fundamental question for kind of like, what is the native fold of a protein, right? If the history of your lineage matters for what form you will adopt, right? And I think, that is sort of the mystery and excitement of of all of this, right? That there clearly is some record of past history that can be stored this way. The question is, how do we understand it, right? Can you control it? How does it get erased? All kinds of things.
Yeah. So I think it would be the last question, because it's already 6:30.
Yeah. You guys have to eat.
It has been great discussion, great seminar. So thank you very much, Dan, for your seminar, for so many good information you gave us. And thank you for these people that was connected in Zoom and in YouTube. And so again, let's thank Dan for the seminar.
Thank you so much.